separated sort of like,
Right.
it's both direct.
It is direct.
But also just
Oh,
and then never random.
Okay, so we're starting a new topic today, and as you can tell just from the slide I've got up there, it's going to be complicated, all right? Of all the topics that we cover throughout the course, signaling as a whole has garnered the greatest number of Nobel Prizes than any other. And if you think about it, there's a lot of reasons for it. Not only is it complicated, but it also deals with just about signaling is integrated with every other cellular process that you can imagine. So it's not terribly surprising that you're going to have all sorts of different laboratories approaching this whole issue of signaling from many, many different perspectives. The interesting thing is that if you notice, if you just kind of look at the areas that these Nobel Prizes were awarded in, most of them deal with the discovery of the molecules that are actually involved in signaling. Growth factors, hormones, nitric oxide down here, one of the more recent ones. So that sort of tells you there that for many, many years, the understanding of cell signaling was basically underappreciated because you didn't know who was doing it. And now we pretty much have a very good idea about who the players are. And as a consequence of that, the focus is shifting now towards the mechanisms. How do things happen? And so, for example, the mechanisms of protein kinases, the mechanisms of G proteins. So there's really been a shift in the emphasis of research on signaling that's moved away from who does it to how do they work. Okay? So we're going to be getting into this, going through a couple of paradigm signaling pathways throughout the unit. But the way we're going to start off is kind of take a big picture overview of going through all the major components that you're going to see involved in signaling and come away with a bit of an appreciation over how do they function, okay? All righty. So, just to start it off, this is kind of the hanging curveball of signaling, is you can define signaling processes very, very simply, just based on the distances that are involved. And what we're referring to are the distances between the source of a signal and its actual cellular target, all right? And you can basically see that it runs the gamut from long distance to autocrine signaling, which means you can do it yourself, all right? So if we just go through these, kind of taking them in order, endocrine signaling is the most common mechanism that you find in multicellular organisms. Endocrine signaling was essential to the evolution of multicellular organisms simply because once the organism gets large enough, cells are no longer in direct physical contact with one another. So it actually required the ability to transduce signals over very long distances. It also meant that the cells in the organism, it gave them a way of being metabolically integrated. So you can have multiple cell types in different tissues or organs. They can all respond at the same time to a given extracellular signal. Okay? Now, endocrine signaling, the molecules that actually mediate the signaling are referred to as hormones. All right? So a generic definition of a hormone is any diffusible molecule that participates in endocrine signaling. That's pretty broad. That basically casts a pretty wide net over who can be identified or defined as a hormone, but it also puts no limits as to what those molecules can be. All right? And as you're going to see, some molecules can actually participate in different types of signaling. Okay? So, but the key thing with endocrine signaling is the source is at a very, very large distance away from the target, and the hormones are typically diffused through the circulatory system or the bloodstream. Okay? Now, something to bear in mind is we're not going to spend any time at all talking about the regulation by which these signals are produced. We're focusing on what happens at the cell, at the target, when it binds to an extracellular signal, okay? This whole area of how you produce, generate, and regulate the production of signals comprises a whole different subset of cell biology that's classified by the generic name of endocrinology, okay? So we're focusing on what happens in response to a signal, not how you go about making it, okay? So endocrine signaling is the long distance runner. If we move things a little bit closer together, now we're dealing with a situation that you're pretty much always going to find in nerve impulse transmission across, for example, a synapse, all right? You also find this in embryonic development, and that's paracrine signaling. Paracrine signaling means the cells are much, much, much closer, in closer proximity. It doesn't utilize a circulatory system or the blood system to transduce the signal, but instead it means the cells are in close proximity, but they're not physically touching. Okay? So we don't invoke a blood vessel here to transmit it. It means the signal just crosses through a space separating two cells. All right? So in paracrine signaling, they're close, but they're not actually hooking up to each other. All right? We can even take it closer. And that is autocrine signaling. Now, autocrine signaling solves a lot of the space and the distance that's involved because in autocrine signaling, a given cell can generate the signal and also respond to it. Okay? Now, this can also be this same cell here that's responding to its own signal that it produces, this could also be an example of endocrine signaling, because there's absolutely nothing to keep this diffusible signal that's being produced moving through the bloodstream and then targeting distant cells. Okay? But autocrine signaling just means that the cell that produces that signal can also respond to it. It means it has to have a receptor for that signal. Where you most commonly find this is in the regulation of cell proliferation in cancer. And most typically what you're going to find is that normal cells require the ability to have a receptor that will only be activated in response to an extracellular signal. And as you're going to see, what happens in cancer is that either the receptor itself or signaling components that work downstream of the receptor are going to have acquired one or more mutations that cause them to be activated even in the absence of an extracellular growth factor. So in fact, the cell is put into a continuous pro-proliferative loop even in the absence of any growth factor that has to bind to the receptor. Okay? The one down here at the bottom, actually, it should go in between these two. Okay? Because it's another example where we require two cells, but they're actually physically touching one another. And the difference with this last particular mechanism that you see in cell communication is that you notice there's no diffusible factor involved. And that is, what you have is that now the two cells that are involved in this type of signaling actually have to be physically touching one another. There's no space there. And what is happening here is that an integral membrane protein actually serves as the source of the signal, and it physically binds and interacts, again, by protein-protein interaction, with integral membrane receptors that are on the target cell. So there's actually a distinction here between this type of signaling and paracrine signaling. Paracrine signaling, you can still have a space. You still have a diffusible molecule. In plasma membrane attached protein signaling, everybody's hooked up directly on the cell membrane itself. So the only way that you can have that type of signaling is between cells that are in basically they're neighbors to one another. And where you're going to see interactions of this sort is predominantly during embryogenesis, where you have changes in cell fate by adjacent cells literally reprogramming each other to generate a new cell type. Okay? So that's an outline, more or less, of the signaling processes. This first unit, we're going to be focusing almost exclusively on endocrine signaling. We'll get down to autocrine signaling later in the course when we're going to be talking about cell proliferation and then ultimately moving into cancer. Okay, two general ways that diffusible signals can get into cells. Okay? The simplest, the least complicated, and for those reasons we're not going to talk about them, are these guys over here. And that's where you have hydrophobic signaling molecules, for example, steroids. Okay? They're produced by a particular cell or tissue. The molecules are secreted, released into the bloodstream, and because they're hydrophobic, we don't need a cell surface receptor. The receptors for these types of hormones, particularly steroids, are actually cytosolic proteins. So we don't need anything on the plasma membrane. So when a steroid binds to its receptor in the cytosol, the receptor changes its conformation and it immediately goes to the nucleus. We're short-circuiting so many steps in the process that what we're going to do is that with this type of signaling, we go straight from the cytosol to the nucleus and we change gene expression. Takes a long time, okay? We're more interested in short-term changes in physiology. So the signaling processes that we're going to be going through are going to be ones that entail small hydrophilic or maybe amphipathic molecules that bind to intramembrane cell surface receptors. Okay? Now, this creates a different problem. This is where the term signal transduction is actually now used and why it's applicable. And the reason is the signal itself is the hormone. It never crosses into the cell. It remains outside the cell bound to the receptor. So the problem that we now have to solve is how do we change the physiology or metabolically reprogram the cell by transducing the signaling response across the plasma membrane. The signal itself remains outside the cell. Because of that, you can think of a hormone or a growth factor as being the first message. The first message is the diffusible molecule that remains outside the cell, and that's going to trigger everything that happens. But inside the cell, what you're going to see is that this type of signaling is now going to involve other small diffusible molecules that are going to be called second messages. The second messages kind of pick up the process that was initiated by the first message outside the cell, and now they're going to relay it inside the cell. So we have a handoff of sorts. We go from a first message to a second message. One of the things that's going to happen during that process, the signal gets amplified. We go from one molecule binding to one receptor to hundreds, if not thousands, of second messenger RNAs, or second messages, sorry, that are going to be generated inside the cell. Okay? Both of these pathways have short-term and long-term physiological effects. What we are going to be focusing on in this unit is short-term. Responses that take place in a matter of seconds to minutes. Okay? And the way that that's going to happen is, again, focusing on this pathway over here on the right. What's going to happen is to make them fast, to make them rapid, and to make them reversible, we're going to be modifying pre-existing proteins. We're not having to make any new ones. That saves us a huge amount of time. So what it means is that the way that we're going to change the activity of proteins that are already in the cell is generally going to be by changing phosphorylation states. So this is where protein kinases and phosphatases are going to become the major determinants in changing cell physiology in the short run. We want to change whatever a cell is doing metabolically very rapidly, for example, in response to the hormone epinephrine or glucagon or insulin. We want to change what the cell is doing very quickly. But as soon as that stimulus is gone, we want it to go back to doing what it did before. So reversibility is a key part of this. However, what we're also going to be moving into, though, as we get down again to looking at cell proliferation, we're going to be looking at long-term effects. Long-term effects are going to entail, yes, we're going to change physiology in the short run. Metabolism is going to change. But in the long run, we're going to reprogram the cell genetically. So genes that are being expressed are going to be turned off, and new genes are going to be switched on. That's a process that's going to take hours. It can even take days. But ultimately, what you're going to see, we're going to focus on the long-term effects when we look at signaling that's initiated by receptor tyrosine kinases. Because those are the ones that are going to be involved in causing quiescent cells to re-enter the cell cycle and to resume cell division. It's also the signaling pathway for most of the mutations that convert normal proteins involved in signaling, called proto-oncogenes. That's where those proteins are going to be converted to oncogenes that will now cause abnormal cell proliferation. So we're going to look at both short-term and long-term. We're just putting the long-term, we're putting that on the shelf for now, and we're just going to be focusing on short-term signaling. These are the main classes. There's 4 classes of cell surface receptor mediated signaling. Okay? And they basically deal with differences in the mechanism by which the binding of an extracellular signal is transduced across the plasma membrane and generates a metabolic response in the cell. Here's the good news. We're only going to focus on 2 of them. One now, one later. The process we're going to focus on in this unit is signaling over here that's mediated by G-protein coupled receptors. And as you're going to see, it's going to mainly invoke the requirement for second messages and protein kinases that are going to alter the activities of pre-existing proteins. We're looking at it for quick-acting, reversible responses. Yes, they ultimately have long-term effects, but we're only going to focus on the short-term signaling. Examples of this type of signaling, we're actually going to look at all of these in one form or another, but we're going to focus predominantly on beta-adrenergic receptor signaling. That's the signaling that's initiated by epinephrine. But we're also going to be taking a look at variations on that signaling process by looking at acetylcholine signaling, adenosine signaling, and glucagon signaling. All of these processes go through G-protein coupled receptors. Okay? If you understand one, you can understand all of them because they're just variations of each other. This is the pathway that we'll get to down the road. That's RTK, or receptor tyrosine kinase signaling. And in this case, what you're going to see is that the receptor itself is a tyrosine kinase. And what happens is that we immediately go off into the kinase race by having one kinase activate another, activate another, activate another. It's referred to as a kinase cascade. One of the biggest differences between RTK signaling that we'll get to down the road with cell proliferation and G-protein coupled receptor signaling is that G-protein coupled receptor signaling requires second messages. RTK signaling does not. RTK signaling is much more direct, and it's like building a jigsaw puzzle. RTK signaling requires protein-protein interactions from start to finish. GPCR signaling is going to require second messages. It does require changes in protein-protein interaction, but the second messages are going to be playing indirect roles that are going to mediate what happens up here at the plasma membrane and what happens down here inside of the cytosol. Okay? So that's just kind of looking forward to where we're going. Why GPCRs? G-protein coupled receptors are the single largest class of integral membrane proteins that are out there. Okay? The human genome encodes probably more than 1,000. There's at least 1,000 G-protein coupled receptors encoded for by the human genome. We still have about 150 to 200 for which we have no idea what they're involved with. And when I say that, what it means is they're described as orphan receptors. We don't really know who activates them. We don't know who the extracellular signals are that actually bind to them. And equally important, we don't know the physiological processes that they are involved with. So there's still a lot to be done in this particular area. But here's the fascinating thing. G-protein coupled receptors are involved in the greatest number of signal transduction events that are out there. They are involved in proliferation, glucose metabolism, muscle contraction, olfaction, taste. So if you look at all the different stimuli that a cell can be exposed to and be expected to change its physiology, G-protein coupled receptors are the go-to players. We've got more of them, and here's what's interesting about them. They all have the same generic structure. So if you ever see an integral membrane protein with 7 membrane-spanning alpha helices, it's a G protein coupled receptor. Okay? You can divide them into 2 functional domains. The extracellular region, which is out here, which is generally comprised of the first 3 extracellular loops. That's where the ligand binds. That's where epinephrine binds. That's where glucagon binds. That's the binding pocket. Binding of those extracellular signals to the extracellular domain of the receptor then changes its conformation so that the action down here inside the cell involves these 2 cytosolic loops, C3 and C4. That's the region that interacts with the G proteins that they are coupled to. So we've got 2 functional domains. Activation on the outside, activation on the inside, ligand binding on the outside. What does this mean? It means that we can make some really cool chimeric proteins here. So one thing to start thinking about, we can take any pair of G protein coupled receptors and make chimeras between them. And the easiest way to think of the chimeras is we can take the first half of a G protein coupled receptor and hook it up to the second half of a different one. So now what it means is that we can swap the extracellular signal that a given GPCR binds to, and secondly now, we can swap who it is going to activate inside the cell. Because these GPCRs then give their specificity for the type of cellular responses they're going to trigger based on the different G proteins that these activation modules interact with. Okay? So again, the idea is, remember, these are bifunctional proteins, and we can physically or biochemically separate who they bind to from the cellular process that they're going to interact. Okay? Here's why they're called G protein coupled receptors. This is the G protein that they're going to be interacting. Now there's a difference here between these types of G proteins and the little ones that we've talked about so far. All of the G proteins that we've talked about so far, RAB, SAR1, ARF, RHO, RAC, CDC42, all of those are small monomeric G proteins. Now what we're dealing with are what are called heterotrimeric G protein complexes. They're heterotrimeric because they've got 3 subunits, and the subunits are all different. Alpha, beta, and gamma. Okay? Alpha and gamma have covalently attached lipid anchors. Beta is just sort of a parasite that hangs out with gamma, so they're always together. Okay? That means that these guys are linked through these lipid anchors into the cytosolic leaflet of the plasma membrane. So when a GPCR finds its ligand, that altered conformation is going to cause it to interact with a given set of trimeric G protein complexes. If you look in the human genome, we encode 21 alphas, 6 betas, and 12 gammas. Guess what? You can put those together in any combination or permutation. So what that means is that our cells has an incredible amount of diversity in terms of what combinations of trimeric G proteins we can put together that can bind to different receptors bound to signals. That's one level of complexity. The second level of complexity then is that these different G protein complexes interact with different effector enzymes inside of the cell. So different G protein complexes can not only interact with different receptors, they can interact with different effector enzymes to change the physiology of the cell. Okay? We're going to simplify things down a bit. We're only going to look at a couple of families of G proteins. The main one up here are what are referred to as GS family. S for stimulatory. The GS families of trimeric G proteins activate adenylyl cyclase. That is an integral membrane protein that's going to synthesize the most important second messenger that we're going to deal with in this unit, and that's cyclic AMP. The other trimeric G proteins that we're going to talk about are called either GOs or GQs. They do the same thing, but you just find them in different cell types. GOs and GQs activate a different integral membrane protein, which is called phospholipase C. And what it does is that that's an enzyme that's going to cleave a particular type of phospholipid. Turns out to be phospholipids that contain phosphatidylinositol. And what phospholipase C does is we're actually going to get 2 second messages at the same time. We're going to cleave off the polar head from the phospholipid, and that's going to give us the second message, IP3, or inositol trisphosphate. The other second message is actually a parafatty acid to stay behind in the plasma membrane, and that's going to be diacylglycerol. You get 2 for the price of 1. Here's what we're only going to focus on, though. When we activate trimeric G protein complexes, alpha is the actual GTPase. Alpha has to be activated, like every G protein, by exchanging GDP for GTP. The receptor does that for us. So the receptor is going to be the GEF, okay? That now means that alpha, when it's bound to GTP, is going to be the main player. And the way it's going to work, as you're going to see, alpha then dissociates from beta and gamma. And it's going to be the alpha subunit itself that activates adenylate cyclase or phospholipase C. It's going to be the actual activator. Something to bear in mind. When alpha separates and goes its own way, beta and gamma stay together, and they go off and do stuff as well. So beta and gamma do play important roles in cell signaling. We're not going to be getting into that. We're only going to be focusing on alpha, okay? So we're keeping this a little bit more simpler than it actually is, because this is the only one of the 3 subunits we're really going to be dealing with. We don't have to worry about beta. We don't have to worry about gamma, okay? A UVA connection here. The person who was the co-recipient of one of the Nobel Prizes in physiology and medicine was on the faculty of UVA at the time he discovered those proteins. His name was Alfred Gilman. He passed away last year of pancreatic cancer. Gilman is the guy who made the discovery that there was something involved in signaling between a hormone receptor and cyclic AMP. He is the founder of G proteins. He was the first one to discover them. And the reason he called them G proteins is he made the following connection. If you take a hormone like epinephrine and treat cells with it, the output that you would get would be an increase in cyclic AMP. Cyclic AMP is generated by the enzyme adenylate cyclase, and its only substrate was ATP. Cool with that? Well, here's what Gilman discovered, is that if you take cells and treat them with epinephrine, you had to add GTP to get the cells to increase cyclic AMP. The synthesis of cyclic AMP only requires ATP. So what Gilman discovered was there's a need for GTP to go between the hormone and synthesizing adenylate cyclase. So he assumed, and rightfully so, there was another protein that was in between the receptor and adenylate cyclase that required GTP. He didn't know what it was, so he named it the G protein. That's where they first got their name. And then he went on to actually discover the trimeric G protein complexes. And they are the bridge They're the intermediary between a hormone receptor and actually increasing adenylate cyclase inside of cells. That's why he won the Nobel Prize. But the UVA connection is he did a lot of that work while he was here in the faculty over in the med school. Okay, here are the second messages. Memorize them. You're going to be seeing them again in different shapes and colors. And importantly, you're going to see that pharmacologically, we can mess these guys up. And a lot of us do it every morning when we come in here because, as you're going to see, you drink coffee, caffeine's a stimulant. It's going to be perturbing the activity of at least one of these second messages. Okay? So they're all pretty simple. Two of the second messages are nucleotides, but they're unusual nucleotides. They're cyclic or cyclic. And that is what you notice is the 5' phosphate forms a cyclic phosphodiester bond between the 5' OH and the 3' OH of either AMP or GMP. So cyclic AMP, cyclic GMP are synthesized by enzymes that are called cyclases. The substrate would be ATP or GTP. Okay? So both of these are synthesized by integral membrane enzymes. Okay? Cyclic A, cyclic G. They're going to be made, if we just go down the pathway here, if we're looking at second messages, how do we make them? Well, in that case, it's the cyclases. If you're over here, here's what phospholipase C does. Phospholipase C takes phosphatidylinositol. So here's IP3. Imagine if that phosphate was originally esterified to that hydroxyl on glycerol. That's a phospholipid. Okay? Phospholipase C cleaves that bond, and this is what generates this little diffusible second messenger inside our cells, IP3. It also leaves this guy behind. It's diacyl, 2 fatty acids that are still esterified to glycerol. So we refer to it as DAG. This stays behind in the plasma membrane. This diffuses into the cytosol. So phospholipase C generates 2 second messages. Calcium is another second message. The major function of calcium inside of our cells is signaling. And the reason is, as you already know, calcium levels are normally very, very low inside the cytosol. If we want to get a physiological effect due to changing calcium, it's very easy to increase its concentration. What you guys are going to see is that the sole function of IP3 when it's generated is to come down to the endoplasmic reticulum and open an IP3 gated channel for calcium. So IP3 is an intermediary to increase intracellular calcium. That's all there is to it. And then finally down here, we've got this other really cool little second messenger RNA that's been described as a neutrino. And the reason is it can pass through any membrane, anytime. It's nitric oxide. Not to be confused with nitrous oxide, laughing gas. This is nitric oxide. Nitric oxide is about the shortest lived second messenger that you're going to encounter. It only exists for about seconds before it's degraded. Okay, so it's very quick acting, very transient. But what you're going to see is the way we're going to divide all of this up is we're going to take cyclic AMP, these two guys over here in calcium. So we're going to bundle those second messages together. Okay? In another signaling context then, we're going to bundle cyclic GMP, these two guys, calcium and nitric oxide. So you're going to see that we're going to take these different second messenger RNAs, put them together in a couple of different physiological contexts so you can see how they all integrate together. What you're going to see as we go through these signaling pathways is that we have to get into how are the second messenger RNAs made, what are their targets once they're there? So in the case of cyclic AMP, it's only got one target. Its target is going to be the serine-threonine protein kinase, pKa. And then we have to get rid of them. So if we want to drop the level of these second messenger RNAs, you don't just want to turn off their synthesis, because now these second messages are going to hang around in the cell. We have to degrade them to drop their concentration quickly. So what you're going to see is, in the case of cyclic AMP, we're going to get rid of it by the CAMP phosphodiesterase. And all that's going to do to inactivate cyclic AMP is to cleave this bond right there. And what it does, it just converts 5' AMP, which is inactive. Here's something you're going to see, is that when a cell is responding to a hormone to change its physiology, while it's ramping up the production of second messages to change their physiology, they are also ramping up the mechanism to degrade the second message. This may seem like a paradox, but it's not. So at the same time a cell is changing its physiology, it's getting ready to go back to its resting state that it was at before the hormone bound. Because it wants to make this change transient, and it wants to make it very reversible. Okay? So that's just something that you're going to see coming down the pipe. All right. These guys here are the most important players in the entire process. Protein kinases. We categorize protein kinases based on the amino acids that they phosphorylate. We've got 3 amino acids that are going to be the targets: serine, threonine, and tyrosine. Those 3 amino acids. If you take a look at the proportion of those amino acids that are phosphorylated, you can see cells go to serine 80% of the time. That's the go-to amino acid. Threonine, a bit lower, 17%. Tyrosine, very rarely, 4% of the time. So serine is clearly the preferred amino acid to phosphorylate. What protein kinases do is that they covalently add the terminal gamma phosphate from ATP onto one of those 3 amino acids. There's 3 categories of kinases that do this. The first up here are called serine-threonine kinases. It's one class, but they can phosphorylate either serines or threonines. It's the largest class of protein kinases. The human genome encodes 400 serine-threonine kinases. There are a lot out there. They're the go-to kinase. Okay? Over here is the smallest number of protein kinases, tyrosine kinases. They can only phosphorylate tyrosine. There's only about 90 of them. And as you're going to see, cells don't like to phosphorylate tyrosine because bad things can happen when it occurs. Tyrosine phosphorylation is primarily going to be used to control cell proliferation. Okay? In between is that we have this kind of an odd nomenclature. You've got a small number of kinases that can phosphorylate all 3, but we call them dual specificity. We don't call them tri-specificity, they're dual specificity. And what these are, it's a small number of kinases that can phosphorylate either serine-threonine or tyrosine. And what you're going to find is some proteins are phosphorylated on both a serine or threonine and a tyrosine. And what you're going to find in every one of these cases is that those amino acids are right next to each other. They might be spaced by one amino acid in between them. So the serine or the threonine or the tyrosine are basically lined up so they can be hit by the same kinase at the same time. Okay? Kinases put the phosphate on. Okay? So we take a substrate protein. The vast majority of the time, the non-phosphorylated protein is biochemically or catalytically inactive. the majority of the time, phosphorylation activates it. So by putting a phosphate on one of those 3 amino acids, you're going to now increase its enzymatic activity. But not every time. You're going to see examples right off the bat where certain enzymes, when they are phosphorylated, they're inactivated, they're turned off. So don't assume that just because a protein gets phosphorylated that you're switching it on. There's going to be proteins that get switched off by phosphorylation. Okay? Kinases switch by adding phosphate. These guys are equally as important. Protein phosphatases have to remove the phosphate from the protein. It's a covalent modification. There's something intriguing if you look at this. We've got more serine-threonine kinases than any of the other two. We've got the least number of phosphatases. So we have more serine-threonine kinases than there are serine-threonine phosphatases. But take a look at tyrosine. This is what emphasizes the fact that cells do not like to phosphorylate tyrosine on proteins. There's only 90 kinases that can do it. But there's at least 100 tyrosine phosphatases. So it means that cells really want to be very careful about what proteins they phosphorylate on their tyrosine. Because of that, they go to great lengths to have phosphatases that will turn them off and reverse it. Something that you're going to see in all of these pathways, okay? It's just a heads up. Every time that we activate a kinase, when we activate a kinase to phosphorylate a protein, we are also going to be inhibiting the phosphatase that takes it off. What we're trying to do in these signaling pathways is think of it this way. We're changing the ratio of the relative amount of a phosphorylated protein to the non-phosphorylated protein. So we activate the kinases to put the phosphates on. At the same time, we turn off the phosphatases to keep the phosphate on. So when we turn off the kinases, we have to reactivate or bring back to life the phosphatases that are going to reverse it. Okay? What I want to wrap up with this morning is, why was this chosen? Protein phosphorylation is the single most common post-translational modification out there. There's a lot of things you can put on proteins to change their properties. Phosphorylation is the go-to modification to change enzymatic activity. As you can see from this article right here, about a third, between a fifth and a third, of all of the proteins that are in a cell at a given time are regulated by altering their phosphorylation. Why was phosphorylation chosen over evolution as the mechanism to regulate enzymatic activity? There was a review or an article, actually it's a paper, but it was a theory that was put together a number of years ago by the Farrell Lab that actually explored this. And what they did is that they looked at the evolution in the active sites of proteins over evolution that acquired the ability to be regulated by protein phosphorylation. And here's what they found. It's very simple and it's very intriguing. Most proteins are going to be activated by phosphorylation, but there are a few that are turned off. And what you find on the small number that are going to be inactivated by phosphorylation is that in the active site of the enzyme, they have a serine or a threonine. And when you phosphorylate it, remember what you're doing when you add a phosphate. It is big, it is bulky, and it is very negatively charged. Serine or threonine are nice and compact. They're polar, but they're uncharged. So in this particular case, if you're going to negatively regulate an enzyme by phosphorylation, what you are doing is introducing a large, bulky, negatively charged group into its active site, and that inhibits it. And that's what you're going to find with the enzyme glycogen synthase. Okay? What happens with the evolution of phosphorylation to activate proteins is you go back in evolutionary time, and what you find is most of the proteins that are now regulated by phosphorylation originally were constitutively active. That means they are always on. And when you look at these enzymes, what you find in common in their active sites is that they always had a bridge between a positively charged amino acid that was generally arginine, that formed an electrostatic bond with a negatively charged amino acid that's generally glutamate. That electrostatic interaction holds the protein into its active structure so it can be catalytically active. Over evolutionary time, what you're going to see is that glutamic acid gets changed to either serine or threonine. We lose the electrostatic interaction. That means that this enzyme now is going to be inactive because it can't adopt its active conformation. So what happens is the way that we take this enzyme and now switch it on is by phosphorylating the serine, the threonine, or in a small number of cases, the tyrosine. So now what you're doing is that the phosphate reestablishes the electrostatic interaction, and now that restores the enzymatic activity. But now instead of the enzyme being constitutively active, it's inducible. So that enzyme is only going to be biochemically active now when it's phosphorylated. We dephosphorylate it, and now it shuts itself off. This is going to be the primary mechanism that we're going to be going through, is that it's this type of evolution in the structure that's going to allow many proteins to be activated by phosphorylation. Okay? So we'll start that off on Wednesday then and take a look at how we get kinases specific.
There you go. Hi, Eddie.